protein modification by members of the ubiquitin family efficiently regulates the activity of cellular factors involved in almost every biological process. Ubiquitin is attached to a target protein in a covalent manner. When this happens, the folding of the protein changes together with characteristics of the modified target, such as its localization in the cell, its capacity to interact with other proteins, its stability, or its activity. Multiple monomers of ubiquitin can modify the same protein in different places, but ubiquitin can also form complex chains with distinct biological meaning. In pathological situations, chains can vary according to the molecular defects observed in sick cells. Defects in the equilibrium of proteins regulated by ubiquitin family members result in pathologies such as cancer, neurodegeneration, inflammation, or multiple infections. This area has therefore become very attractive to pinpoint biomarkers for disease identification and drug discovery. Detecting ubiquitin chains and protein targets in the context of a disease is crucial to understand the molecular events affected in a given pathology or in response to a treatment. Our molecular traps enable to detect, isolate, and identify key proteins from cell cultures, various biological models, but also from patient samples. There is no need to transfect or genetically modify the biological samples to capture endogenous proteins. The prototype traps captured ubiquitylated proteins, but this technology has been expanded to other ubiquitin family members, such as SUMO, LC3, ATG8, or NED8 molecules. We have developed applications with these molecular traps, including affinity columns that can be used to detect proteins of interest by Western blot, but also to identify proteomes using mass spectrometry. Other applications include the detection of molecular signatures associated with a disease, or high-throughput screening methods to identify new therapeutic molecules. Molecular traps are based in the multiplication of binding domains that specifically recognize a particular ubiquitin family member. The tandem disposition of binding domains enhances the affinity for the recognized moieties, and in some cases offers protection from demodifying enzymes and from the proteasome. All these properties allow to obtain a high purification yield of the proteins modified by ubiquitin family members. With all these characteristics and applications in mind, you can use and adapt our molecular traps to the experiments you are designing. Thanks to their great versatility, you can expand the use of these tools up to the limits of your imagination.